Welcome to MOOC course on Introduction to Proteogenomics. Till this week, you have heard Dr. Mani, Dr. Ruggles, Dr. Bing Zhang that why study of mutations and gene sequencing is so important to understand the clinical conditions. In today's lecture, Mr. Abhijit Dikshit will talk about droplet digital PCR. He will talk about the 3 generations of PCR: quantitative PCR including the normal standard polymerase chain reaction, relative quantitative PCR including the real time PCR, and absolute quantification which includes droplet digital PCR. He will also talk about the basic principle and workflow of the technique. He will then talk about how partitioning of templates facilitates the finding of rare mutations in the gene with normal PCR due to the low abundance. He will also talk about how this technology platform enables us to study the rare molecules even after the presence of the inhibitors. So, let us now welcome Mr. Abhijit for his today's lecture.
My name is Abhijit. I work as a product specialist for Biorad looking at the genomics portfolio. I know it's a really taxing and a long session you guys have had dealing with this part, right. It would be my effort to drag you here, right. I would sincerely try to get it through. If there is anything that is not clear or you guys are not able to take it up, please do let me know. It will be my pleasure to clear it up for you. Yeah. Okay. So, here we are today to talk about a technology called as the droplet digital PCR. As I mentioned, I guess most of you know what at least a PCR is. Okay. So, everybody here, I mean they know what a PCR is. This is what we do. The PCR has generally evolved over technologies. Okay. The first generation of PCRs where you put up a PCR to know a presence or absence of a particular gene, you have to run a gel, right. A presence of a band would indicate a presence of a particular gene. A band is absent, that means the gene is not present. Okay. So, that old traditional PCR is called as a qualitative PCR. You know the presence or absence of a particular gene. Second, as I went a step ahead to let you guys know what a real time PCR is. Okay. A quantitative PCR, if you want to know the amount of DNA or RNA expressed for a particular gene, right, you can go around and do a quantitative PCR. But for those of you who have done real time PCR, you know it is a relative quantification. To know the amount of nucleic acid, you guys have to run a set of standards or run a housekeeping gene and then you compare it with that expression to know the quantity of DNA or RNA that is present. So, that is why it is called as a relative quantification. What we have come up with now, which is called as the 3rd generation of PCRs, is the droplet digital PCR. The major difference being it is an absolute quantification, so that you do not need to run any set of standards or housekeeping genes to know the amount of your nucleic acid in your sample, right. In my presentation through, I will brush upon what the technology is, how does it work, how is it unique from the previous PCRs, right, and what are the advantages. Again, for those of you who feel this technology is new, it is not. The first publication which had come with digital PCR was way back in 1999, but then it has been over 1.5 decades that took for the technology to get evolved and reach this level where it is today. Okay. So, what happens generally in this technology? I mean obviously for all of you have put a PCR, know what a PCR is. We make a same reaction mix, right. This reaction mix will have your nucleic acid, let it be a DNA or RNA, your specific primers or probes, your enzyme, right. We make a same thing here as well, but what we do is instead of putting this reaction for PCR, we split this 20 microliter reaction into 20,000 droplets. These are all in water emulsion droplets, right, and then we put a PCR for those droplets and read those droplets individually. So, instead of one reaction which you put in a PCR, now you have 20,000 reactions going together. That is going to give you 20,000 times more specific and sensitive data as compared to a real time PCR technology. And again, the data that you get here is directly in copies per microliter of your DNA. So, you do not have to do any manual calculations to get to a data. The software automatically analyzes and gives you the precise amount of nucleic acid in your samples. Right? This is just an analogy to compare what a real time or a normal PCR versus this PCR is. This is a clock, right? All these, both of them mean the same. But what you see here is the time is somewhere around 8:40, right? But once you go here, this is the digital result that you get. A more in-depth, a more precise, a more detailed analysis of your data. This is what a digital PCR or a droplet digital PCR does as compared to the traditional PCR technologies. As I mentioned, the main principle of this technology is partitioning. You have one reaction otherwise which you put in a normal or real-time PCR, which is partitioned into 20,000 reactions, and then each of these small droplets works as a small microreactor and undergoes a PCR individually. So that's the main principle on which this technology works, partitioning. And the benefits that you get out of it is, as I told you, it's an absolute quantification, you get high precision and high sensitivity. I'll explain you how and why this happens. So this figure is a very simplistic figure from one of our publications where people have used our technology. Now imagine you are looking for a particular mutant, a mutation, right, which is 40 mutant molecules across your wild type molecules of 40,000 in a particular sample, all right. So that accounts to 0.1% of your mutation, which by a real time PCR or any other technology is practically impossible to detect, because you are searching for 0.1% of your desired product out of the 99.9% of your wild type. What happens because of partitioning is, now when you split this sample into droplets, right, so each of these 40 mutant molecules would go into droplets, and then each of these droplets will undergo a PCR individually. Okay? So now this 0.1% chance of capturing your DNA becomes as high as 33%, because each of your droplets undergo individual PCR reaction and is scanned individually. The data is individual droplet wise. So that's how the percentage and the precision increases for this technology. Okay? This is just a comparison that I have put up. I am sorry the slide is very busy. As a real time PCR versus this technology, the first difference is, yes, this is an absolute measurement, no relative quantification. This is ultra sensitive, where you have to precisely pinpoint and detect the copies of your DNA or the mutation in your particular sample. Even if your sample has a lot of inhibitors, the technology is good enough to capture your data, which otherwise a normal traditional PCR would not. Most important point here is LOD and LOQ. What this means is the limit of detection and the limit of quantification is exactly the same in this technology. Okay? Otherwise, in a PCR, in a real time PCR, they differ. What you can detect is the limit of detection, and what you can quantify amongst that is the limit of quantification. So this technology exactly counts everything that is amplified and gives you the precise data. So the major advantages, if I talk of this technology, would be it's an absolute quantification method. You can go up to a single copy resolution if you want to look at any of the DNA or RNA in a particular sample. You don't need to run any standard curve, so it's an absolute quantification. You can detect your sample in a complex background as well, even if there are very high amounts of inhibitors in your sample or your DNA is not pure, right? The system will precisely still amplify and give you the desired amount of your DNA. Can anyone tell me why would this happen? It's a simple logic that is implied. Why would this technology still help you even in a complex background? That means even if the sample has a lot of inhibitors or something, why would this technology still give you precise data? Perfect. The main principle of the technology, partitioning. So when you partition your whole sample, even your inhibitors are getting partitioned. So the inhibitory effect of your inhibitors is not there. Okay, is everybody clear? Anybody has any queries on this? Okay. And last, this technology is good enough to run with a dye-based chemistry or a probe-based chemistry. So I don't know if everybody is aware, there are two major chemistries by which you can run a real-time PCR. Correct. Cybergreen and Tachman probe, the examples. So Cybergreen is which chemistry? Dye or probe. Correct. A dye-based chemistry, and Tachman is a probe-based chemistry, right? So you can run your samples with either of the chemistries in this sample, right? This is how the workflow goes. You first, as I mentioned, we first generate the droplets, we do a PCR and we read. I'll go through each step in details. This is just a workflow. So in gist, if I have to explain this technology, it would be it expands upon the most proven digital PCR technology with unmatched precision and sensitivity in absolute quantification with the flexibility of using either a dye or a probe based chemistry. So, that is in a gist what the technology principally works on. Now, this is a workflow. We will look at each step in detail. The first thing which I mentioned, you are partitioning your sample. You have a reaction mix, which otherwise you make the same thing for your real time or normal PCR as well, right. This is a cartridge which we have. As you can see, there are 3 rows, right. In the middle row, you put your reaction mix that you have made with your DNA, primers, probes and your enzyme, and you add a oil here in the bottommost part, and you put this cartridge in a droplet generator. This is a droplet generator. So, what the system does is this. This is the actual video of how droplets are being generated. It combines your reaction mix along with the oil, and it generates these uniform size 1 nanometer volume droplets, right. And now these are the droplets which we put for PCR under a normal optimized condition which we would have done for a normal PCR, and these are red. So, the reader here is like a flow cytometer. Okay. What it will do is, this is a probe. The probe will enter each of your well where there are 20,000 droplets. Each of the droplets will be pulled by vacuum, right. They will pass in front of a 2 laser system. There are 2 lasers inside, and it will detect for a positive signal or a negative signal out of each droplet. So, now there are 20,000 readings from one sample, which are then combined together and you get a data. Alright. So, this is how principally it works, and the data that you get is something like this. For those of you who have worked on a flow cytometer, you get a scatter plot, right. So, this is a similar scatter plot that you get. This is a data from one sample. So, each of this dot that you see here, each dot corresponds to one droplet. So, there will be approximately 20,000 readings from one sample. The droplets which are amplifying something which has a DNA that is amplified will have some fluorescence emitted, and they will fall here in this region, and the ones which do not have any amplification will fall here. So, your y axis is the fluorescence amplitude, right, and the software then counts each of them individually and then gives you a data. And this is for your first channel. Similarly, if you are doing a multiplex assay, if you are using 2 different fluorophores, you have a similar data for your second channel as well. So, these are also droplets which do not have any fluorescence, which have no DNA which is amplified, versus these are droplets which have your amplification of your DNA. Okay. Similarly, we do get a 2D plot as well for this, where these are the first channel positive data, for the second channel positive data. This would be for the droplets which have both of your DNAs if you done a multiplex assay, and these are your negative droplets. Okay. Am I clear till now how the technology works, right? Next, what I will show is I have a small video which explains everything that I have told till now in a graphical manner. So, in case there is anything that is left out, you guys can understand it further better. I doubt if the audio system is working. Okay, I will just explain you along as it goes. वॉल्यूम फुल है, यहाँ पे आएंगे डिस्प्ले में पे। Okay, so I'll explain you as it goes. There is no audio coming through, right? So this is how your reaction is split up. These are droplets which will have your reaction mix along with your super mix, enzyme, DNA, RNA, primer probe, whatever. And as it undergoes a PCR, the primers would bind, there would be amplification which is happening. If there is an amplification, the fluorophore is emitted and there is a fluorescence, right? And the ones which do not have any amplification would have no fluorescence. And the system scores each of these droplets individually for a fluorescence versus non-fluorescence. So this is when it's undergoing your thermal cycling, the PCR. Few of the droplets will have amplification, the ones green will have your amplification, the others won't. So that's how the workflow goes. What I was explaining, you have a reaction mix which is prepared. This is the cartridge, you add your samples in the middle row. And the oil goes in the bottom row of each cartridge. You add your oil and you put this in a droplet generator, which will now make droplets from each of these wells. That's how this is the actual video of it makes your droplets. Once they are done, with a normal pipette you take these droplets and then put it in a normal PCR plate and put the plate for PCR. Reaction will undergo PCR, then you remove that plate and put it in a reader, the droplet reader. Now each of these wells, wherever you have added your droplets, this is a probe which will enter each of these wells. This is how it happens. It takes in all your droplets, right, which goes here. There is a laser system here, two-way laser system, which is now going to detect your fluorescence. Whichever droplets have your amplification will be detected positive, the ones which do not will be detected as negative. And that's how the data is being quantified. So that's a real-time reading how it comes for each of your droplets as they pass through.
I hope today you have learned how droplet digital PCR or ddPCR is 20,000 times more sensitive and provides you very specific data as compared to the real time PCR. Advancement in software provides very accurate concentration. Therefore, you need not to do DNA quantification separately. I hope you also learnt about LOD, which is limit of detection, and LOQ, which is limit of quantification of this technology. The next lecture will be the continuation of droplet digital PCR by Mr. Abhijit. Thank you.